Hello, I am Areeb and I teach IB, HL and SL level biology. So today we are going to briefly discuss a class of biomolecules or a category of biomolecules called nucleic acids. And within this class, the different types or the different subcategories that are present. So to start off, what are nucleic acids? Just how proteins, lipids, and carbohydrates all are separate classes of biomolecules, similarly, nucleic acids have their own category of biomolecules. Or we can say that they are a separate class of biomolecules, and within this category are two different subcategories or two different types of molecules present, namely DNA, which is short for deoxyribonucleic acid, and RNA, which is short for ribonucleic acid. And both of these molecules have very essential functions in all life forms. So, starting off, so what is the DNA? The DNA is actually a double-stranded molecule which is made up of DNA nucleotides, which serve as the building blocks of the DNA molecules. Now, the DNA molecule has a very important function, which is to carry or contain all the genetic information of an organism. Now, it is this genetic information which gives the organism its specific features and characteristics, and it is this genetic information that is passed on from one generation to the next. Now, coming to the ribonucleic acid or the RNA molecules, these molecules are made up of RNA nucleotides, and these RNA molecules are single-stranded molecules. So the building blocks of RNA are the RNA nucleotides, whereas the building blocks of DNA were the DNA nucleotides. Okay, this difference is very important. Now, talking about the function of RNA, so the RNA has a very essential function in the human body. So the RNA is involved in the protein synthesis process or is involved in the making of proteins. The RNA molecule uh, basically reads off the specific codes present in the DNA or the information present in the DNA. It interprets it and it translates it and finally makes it into a protein that can be used by our cells. Okay, so now we discussed briefly what DNA and RNA are. Now we are going to look into detail uh, the specific building blocks of both of these molecules, which are the DNA nucleotides and the RNA nucleotides. So as we can see in front of us, we have two structures which are of the DNA nucleotide and the RNA nucleotide. So as we can see here, uh, both of these molecules look pretty similar to us, right? But there are some basic differences which we'll figure out by the end of this video. So to start off, both of these molecules or both of these nucleotides have three basic components that make them up. So looking at the first component, it is the phosphate group, which is enclosed in this oval pointed to by the red arrow that I have just made. So it is this oval and it is this molecule enclosed in this oval which we call the phosphate group. And this phosphate group is the first component of the nucleotide. And this phosphate group is present in both the RNA nucleotides and the DNA nucleotide, and it is similar in both molecules. Now, coming to the second component is the 5-carbon pentose sugar called deoxyribose in the DNA nucleotide and the ribose in the RNA nucleotide. Now, the name ribose and deoxyribose is pretty intuitive, as we can see that the name DNA has the name DNA opens up to deoxyribonucleic acid, whereas RNA is short for ribonucleic acid. So similarly, the DNA nucleotide has the deoxyribose, whereas the RNA nucleotide has the ribose sugar, as we can see here. Now, what is this ribose sugar? This ribose sugar is actually a 5-carbon sugar, meaning there are 5 carbon atoms present in the sugar. This is the first one. This is the second one. This is the third carbon. This is where the fourth carbon is present. And this is the fifth carbon. Now, yeah. So now these intersection points where I've mentioned the numbers are where the 4 carbons are present, whereas the 5th carbon is present outside this outside this sugar ring. Now, moving on. So this is the second component of the nucleotides, the deoxyribose and the ribose sugar. Now looking at the third component is actually called the nucleotide base in both the DNA nucleotide and the RNA nucleotide, which I've just circled right now. In DNA, so per nucleotide, there's only going to be one base, and that one base can be any one of the 4 types that I've mentioned here, which are adenine nucleotide base, thymine base, guanine base, and cytosine base. Whereas in RNA, there's the adenine base, uracil base, guanine base, and cytosine base. Now, there's one base that I've highlighted in both the DNA and the RNA nucleotide. As we can see that the adenine, guanine, and cytosine bases are present in both the DNA nucleotides and the RNA nucleotides. But the thymine base is specific to the DNA nucleotide, whereas the uracil base is specific only to the RNA nucleotide. So that's one major difference between the RNA nucleotide and the DNA nucleotide. So we have just discussed the 3 components that make up the nucleotides: the nucleotide base, the pentose sugar, which is deoxyribose in DNA and ribose in RNA, and the phosphate group, which is common to both the nucleotides. Now there's another difference that is present in between these 2 nucleotides, and it has been highlighted by me in yellow. So as you can see by the arrow that I'm making just now, so if you notice on the second carbon of the DNA nucleotide, there's a hydrogen atom, okay, which I've highlighted. But if you come to the second carbon of the DNA nucleotide, we can see that there's an OH molecule present. So that's one more difference. In the DNA nucleotide, on the second carbon, there's no oxygen with the hydrogen, whereas in the RNA nucleotide, there is an oxygen with the hydrogen. And this again is also intuitive by the names, as the name DNA is short for deoxyribonucleic acid. The deoxy means that it is short of one oxygen, or it is short of one oxygen atom, whereas RNA simply stands for ribonucleic acid, so it does not hint towards any loss of oxygen. So meaning in the RNA nucleotide, the second carbon has an OH group with an oxygen in it, whereas in the DNA nucleotide, the second carbon only has an H group and no oxygen group, or no oxygen atom. So we've discussed 2 differences between the DNA nucleotides and the RNA nucleotides. One difference was the thymine base being specific to the DNA nucleotide, and the uracil base being specific to the RNA nucleotide. And the other difference is that the second carbon in the DNA nucleotide only has an hydrogen group and no oxygen present, whereas in the second carbon in the RNA nucleotide has an OH group present with an oxygen in it. Okay, so these two are 2 of the differences that are present in the in between the 2 DNA and the RNA nucleotides. Okay, moving on. Now, as we can see, now we're going to look at the full picture of the DNA and the RNA molecules. So as we can see in front of us, we have 2 DNA and the RNA strands. As we discussed earlier, that the DNA is a double-stranded molecule, as we can see in front of us, whereas the RNA is simply a single-stranded molecule. And both of these molecules are made up by their own DNA nucleotides, and next to them are mentioned their specific nucleotide bases, 3 of which are common to both, but out of them, 1 is specific to each. For example, thymine is specific to DNA, whereas uracil is specific to RNA. Hope you enjoyed the video. Thank you.